and that is antibody generation and activation. So let's have a look at today's topic, that is B cell ontogenesis. Let's move on. Next slide. Now, by the end of this discussion, you should be able to explain the development and the maturation process regarding the T cells. How do they originate? Where do they come from? And what goes in terms of their development or ontogenesis? Then the surface antigens. Now, some people might be asking, do they have antigens? Yes. When you look at all the immune cells and even other body cells or somatic cells, there are different surface markers, and those are the ones that are used for identification purposes. So even for B cells, we have these surface markers like the CD22, CD19. These are just surface antigens that are used for identification. And for that reason, we usually have monoclonal antibodies if you want to stain specific immune cells, whether it's T cells or B cells or any other cell of interest. We have monoclonal antibodies that are very specific to those cell surface markers. So that is the context that I'm talking about, the surface antigens regarding the B cells. Now, you also need to understand the process through which these B cells recognize their antigens. In other words, we have the B cell receptors, which they utilize in terms of immunoglobulins, and these are very important for recognizing antigens. Then the functions of the B cells, and lastly, we look at the defects that are associated with B cell dysfunction. And that relates to overall dysfunction, looking at the B cell is not properly developed and the functions that comes with it are hindered or impaired. Next slide. Now, differentiation of a specific blood cell type, that is the B lymphocyte, to produce antibody action or cell injury. So these B cells have very important roles because the production of antibodies has various functions as we see them in terms of dealing with pathogens or cell injury. Now, these B cells, they have immunoglobulins on their surface, what we call B cell receptors. And this, from the gene level, all information is stored in the DNA regarding that B cell that is to be formed, or any other cell, or any other function or protein within the body system. This information comes from the DNA. So that particular gene that is responsible for expressing the antibodies, we call them antibody genes. They are not readily encoded in DNA. And we know very well that there's a lot of things that happen in the process of gene expression, because DNA is just stored information, but this has to be decoded into through the process of transcription, then from transcription to translation, and then you have your functional protein, which carries a specific function. So in line with that, then also the antibody genes are within the DNA itself, but they are not yet active, even though they are encoded. And this has to be undergo a certain process, what we call rearrangement or assembled, and then only the functionality of that particular gene or antibody can be expressed. And this process happens not only with the B cells alone, but also with the T cells, because these two cells, they share the same phenomena of gene rearrangement through either T cell receptors or the B cell receptors. And that is the beauty of these immune cells, because that gives them the capacity to identify diverse group of antigens by this process of gene rearrangement. Next slide. Now, from the hematopoietic stem cell, which is the master or the matter of the origin of these immune cells, it gives rise to lymphoid progenitor, and also there is the other one, myeloid progenitor, which gives rise to other granulocytes. But we talk about the lymphocyte progenitor, which gives rise to three cell populations, that is the B cells, the T cells, as well as the natural killer cells. And our concentration now, we are looking at the B cells. Now, this lymphoid progenitor can migrate from the bone marrow to the thymus, and this is regarding to the prothymocytes, which go into the thymus region or gland for purpose of T cell development or ontogenesis. And I know that one is already covered previously. So the B cells don't migrate from the bone marrow. They actually complete the process of maturation within the bone marrow. Okay. So in terms of the stages for this B cell development, we have 4 stages through which these B cells attain its maturity. And we can list them as pro-P stage, the pre-P stage, immature B stage, and lastly, the mature B cells. Let's move on. Now, if we look at the B cell development, we have said that these B cells have various functions, but as the B cell begins the process of development from the germline or its native DNA, we want to see what are some of the factors that are very fundamental regarding its development. So number one, there must be the presence of bone marrow stromal cells. That is a kind of fibroblast that provides cell-mediated contacts and the secretion of cytokine interleukin-7. So the presence of these stromal cells is very important because it provides certain functions that helps the B cells to begin the maturation process. And we have seen it provides contact. That is one important thing. Then also it secretes interleukin-7, which has a role to play in terms of the B cell development. So in a nutshell is that this interleukin-7 must have a receptor, right, on the B cell, the native B cell that is beginning to be formed, so that that interaction can be effected. Number two, there is also the rearrangement of the immunoglobulin antibody. Remember, these immunoglobulin genes, they are already existing, as we have said, within the DNA itself, but they are not yet functional, and that requires the rearrangement of the immunoglobulin antibody genes. And you can say, I give this example, if you look at the bricks, you can have different shapes in terms of rearrangement to bring out a certain shapes. And that is synonymous with what I'm talking about, the rearrangement of the immunoglobulin genes. So this happens in the pro-B stage. And this is where these genes begins to rearrange. And finally, we get to the mature state of the B cell. But one thing I want to mention here is that the rearrangement begins with the heavy chain and then with the light chain. And we'll see this one, especially from the diagrams that we'll see shortly. So what are these immunoglobulins? We say antibodies, but remember there's no difference between antibodies and immunoglobulins. When we talk about immunoglobulins, we have the issue of structure and the function in mind, but antibodies is just a general term, and that's why we talk about subclasses or classes of immunoglobulins. So we talk about immunoglobulin M, immunoglobulin D, immunoglobulin E, and immunoglobulin A. So if you talk about immunoglobulin, that remember you are referring to a specific functionality of an antibody in form of immunoglobulin. but antibodies is just a general term. So then another requirement is the expression of a particular cytokine receptors and the transcription factors that can help the B cells to develop further. Next slide, please. Let's move on to the next slide. Now, let's have a glimpse of the antibody or immunoglobulin itself. They have this common structure, the V-shaped structure, where we have the arm and the stem. So we have each of and every immunoglobulin has 2 heavy chains and 2 light chains. And we have the variable regions and the constant regions. Now the variable regions these are the interacting sites with antigens. We call them antigen binding sites, where the antibody can bind or have contact with an antigen, and that's why we call them antigen binding sites, or AP. Then we have the constant region or the stem, which normally binds with complement, especially when it comes to issue of phagocytosis, where the participation of complement is very key for expedited phagocytosis or opsonization process. Now the B cell receptor, BCR, is an immunoglobulin that you find on the surface of the B cell. So this immunoglobulin has to go through the process of recombination or gene rearrangement, and that gives it's now the proper way that it can interact with antigens. So you can see there we have the heavy chain as well as the light chain. And then, like I've said, this constitutes the immunoglobulin structure, and we have the antigen binding sites, and also we have the biological activity or the stem site of the immunoglobulin. Next slide. Let's move on. Next slide, please. Now, this leads us to the process we are talking about, the B cell development. Now, we are looking at the earliest stages of B cell development. And we have mentioned some of these factors before, that we need the presence of the bone marrow stromal cell for other purposes of providing contact with this B cell that is in development. Now, the bone marrow stromal cells provides these 2 important functions, and as we have said before, it provides the contact to B cell progenitors, and this is regarding to the adhesive interaction through which the integrin, that is the very late antigen 4, which is very key for binding the B cell, and also the vascular cell adhesive molecule 1, which is important in terms of link adherence. So the very late antigen 4 is very important for movement of B cell from one location to the other, as well as through the inflammatory process. Remember, during inflammation, you need more of the white blood cells to participate. So, and that is the time that more cells are actually generated or produced from the bone marrow as per the requirement. So that very late antigen 4 provides that inflammatory response as well as activating the proliferation process, while the vascular cell adhesive molecule 1 provides the adhesive for the B cell so that it can undergo proper orientation in terms of development. And that requires some hours or some days for this process to go on. Next slide, please. We also said it requires the function of the cytokines, and specifically the stromal cell provides the interleukin 7, which is secreted by these bone marrow stromal cells, and it binds with the receptor that is expressed on both the pro and the pre-P cells with the function to stimulate the proliferation of B cells. So the stromal cell provides the interleukin-7, which has its receptor on the pre-P as well as the pro-B cell, B cells, that once it binds, it activates that cell to begin proliferation into another form of the B cell with better functionality in terms of moving forward. Let's move on. Now, this gives you kind of a summary of what happens, and I have to reiterate, we have mentioned that there is this existence of immunoglobulin genes that are not yet rearranged, but beginning with the stimulation by the stromal cells, providing contact and secreting the interleukin-7, then this process kind of causes this B cell to begin rearranging its immunoglobulin genes, and this will begin with heavy chain, then comes to the light chain, and finally you can find it like there is now a conjoining of these chains, heavy chains as well as light chains, to make an immunoglobulin form of a structure that provides the shape of a B cell receptor that you can find on a mature B cell. So in a nutshell, we can talk about the production or the generation of the B cell receptor on the surface of a B cell that never existed before. Let's move on. Let's move on. Now, this gives you the overview of what we have talked about. Now, if you look at the early pro-B cell, we have talked about the rearrangement of the heavy chain molecules or chains. And what you see here, basically, we're talking about the VDJ chains that need to be rearranged. And this is done by the participation of RAG1 and RAG2 recombinant activating genes, which are very important proteins for bringing out this rearrangement. Now, in large pre-P stage, you can see even the B cell receptors beginning to appear, but they are not yet mature. So the main key here is that unlike the pre-P, immature and mature B, where you can see the expression of the B cell receptor on the surface, in the pro-P, you only have the rearrangement of genes, but no expression of the B cell receptor on the surface. So that is the major distinction that you can make at this stage. Next stage, please. Next slide. Now, if you look at this, probably it gives you a better way of you looking at it. We have the pro-P stage where you have the heavy chain rearrangement, and I've talked about it before. But again, look at the pre-P, you have the light chain rearrangement. And like I said before, from the pre-P stage to mature B, you can always see the expression of the B cell receptors on the surface. And that finally leads to the production of a mature B cell. So the other thing I want to mention here, look at that the pro-P and the pre-P stage, this happens in the bone marrow, while the mature P cell that is fully functional, it is released into the periphery or the peripheral circulation. So if you probably want to look at the surface markers, the B cell, if you are interested in mature B cells, then you can look at certain surface markers that you can find at any stage, because some may be expressed early in life, but when the cell matures, they get removed or they are eliminated while the others are required at the time of maturity, like the B cell receptors or immunoglobulins that you may not see in the earlier stages. And this gives you the better way of differentiating between the mature and mature B cells. And this can be applicable even to T cells and the other immune cells. So in pre-B cells, the rearranged immunoglobulin heavy chain, as we have talked about, pairs with other proteins like the mu5 and the pre-P proteins, which are the surrogate light chain markers. And this forms the pre-P cell receptor, which we have talked about as the native B cell receptor that begins to appear on the surface of the B cell. And this always signals that the cell needs to proliferate or move further to another stage. Next slide, please. So in summary, we are looking at what happens at each and every stage of the B cell development. Now, this table gives you a kind of a detailed summary in terms of the heavy chain, which happens in the stem cell or the germline. And when you move to early pro-P stage, we have the decay chains rearrangement. And then late pro-P, you can have the further rearrangement of this, and the process keeps on moving as the table indicates. So the light chain, you can also see the comparison where it begins, and then the surface immunoglobulin, which we have said, actually begins to appear from the pre-P stage till the maturity. You can have those. Then you can have the dependence on this stromal cell, whether they are key in which process, and that gives you the contrast where the interleukin 7 is dependent or is not required. So that is a very good summary of what we have just talked. You can look at it keenly and see what happens at every stage in terms of acquisition of these receptors. Next slide. This is a very critical slide because it is synonymous with the T cell ontogenesis. Now, in early pro-P cell, we have mentioned about the heavy chain gene rearrangement. And what happens, there is participation of the two proteins, the mu5 and the Phi3P proteins. And what happens from this stage, they must go into the next level of checkpoint, that is pre-P cell receptor, whereby if the cell does not, that is the B cell, does not express the pre-P cell receptor, then it receives the signal for apoptosis. And this is the first checkpoint. So the next level is the rearrangement on the light chain, which now gives rise to alpha, gamma, immunoglobulin gamma and immunoglobulin beta chains that are formed. And this one also has to go to the second checkpoint, whereby the B cell receptor has to be checked whether it's properly developed. If there is no B cell receptor, then again, this cell receives a signal to die through apoptosis. So then the next level is just to mature into a functional B cell. So these are the two levels whereby the B cell has to go into check before it finally goes into blood circulation or the peripheral region. Synonymous with the T cells, you have the first checkpoint whereby there's a positive selection, where you find like the positive CD4, CD8, that's a double positive, has to interact with the self antigens, but the signal has to be low, not very high. So those that show some level of recognition or interaction with the self-antigens must be allowed to move from the medulla region, from the cortex region to the medulla region. And then finally, there's the second signal of negative selection, whereby those that are highly selective, highly binding with the self-antigens, they have to be removed. And those that are less or no showing any reactivity, then they can move to the blood circulation. So just like the the T cells, also the B cells have the two signal of checkpoint, so that you ensure that only the cells that are not reactive to the self-antigens are allowed into blood circulation. Next slide. Now, the process of B cell is not 100% effective. Things can change or can occur somewhere. And one of the defects that we can have, or deficiencies in terms of B cell development, is the development of this Pratton's tyrosine kinase disease, which is encoded by the gene on X chromosome, which is very essential for B cell maturation. So in the event that this enzyme, or the Pratton's tyrosine kinase enzyme, is not properly expressed or functional, then you can imagine the process of B cell development is hampered or is compromised. And in that case, you have people, this particular case, where they don't have the production of antibodies. Because of this gene mutation, then the process of B cell maturation stops at the previous cell stage, and this results in a disease called Pratton's X-linked gamma-globulinemia, whereby there is no production of antibodies. So what happens? Such a person or patient may experience recurrent infections, especially with cyanopulmonary infections caused by Staphylococcus pneumonia or Haemophilus influenza. So these are some of the prognosis features that somebody can employ, especially if it's a child that faces these recurrent infections. Of course, there are many other factors that can cause that. So you can even check the immunoglobulin profile within the serum and see what happens, because there's always the normal and the abnormal results that you can use to predict what happens. It's not straightforward of knowing that, oh, this person with recurrent infection has this. Of course, it has to be informed by many other factors. Next slide. Now, let me mention here that there are two subsets or populations of the B cells. We have the P1 type cells. These are the minor subset, and usually you find them in early embryonic life with unknown origin. So people say they originate from the liver, but the textbooks are a little bit unclear about their origin. But what is important about them, they are self-renewing, and they are usually the primary cells you find in the pleural or peritoneal region, and they produce polyspecific antibodies. The contrast from polyspecific, we have monospecific. So polyspecific meaning that can cause some level of cross-reactivity when the antigen, the antibodies share similar antigens. It can cause cross-reactivity. Now we have also the B2 cells, which are the major subset that develop after birth, and usually they express the CD5 glycoprotein, which are repressed from the bone marrow, and they perform the primary location. They are found in the lymphoid organs. I know you know what lymphoid organs. We have the primary and secondary. The primary, we talk about the bone marrow, and then secondary, we have the peripheral, like the lymph nodes, where including the spleen, where you have majority of these resident B cells, whether they are in native form or in memory form. Okay, so these B cells, the P type 2, are very important in generating very specific antibodies, or you can talk about monoclonal antibodies for that reason, because they depend on the T cells for activation. While the T cells can recognize antigens in their native form, the T cells don't, because they require an antigen MHC, a complex, for them to be activated. So whenever the T helper cells activate B cells to produce antibodies, those antibodies are highly specific. Okay, next slide. Now, if you may want to recap what we have just talked, you can ask yourself these questions: the functions of the B cells, which molecules form the B cell receptors that we have talked about. So let me mention here that the B cell receptors are actually immunoglobulins that are found on the surface of B cells, and any mature B cell must have immunoglobulin M on its cell surface. Now, what happens in the native form? Sometimes there is co-expression of immunoglobulin B with this B cell. So you can have the immunoglobulin M as well as immunoglobulin B. So that is in its native form. But when the B cells get activated, of course, they transform into a plasma cell, which is a bit larger in size and rich in energy, and this can differentiate into antibody molecules whenever a need arises. And the function of these antibodies, you can find out in textbooks, but they can bring about neutralization, they can bring about opsonization, and these are some of the applications of um, antibodies. You can find out the functions of antibodies as you read it through your textbooks. Then how do B cells recognize the antigens? We have seen that these B cells must first go the process of gene rearrangement, which is even the importance of gene rearrangement above there, and then they acquire mature status by having the B cell and the IgD, which must interact with antigens. How do immature B cells produce? Do immature B cells produce antibodies? We can say no, because you only need mature B cells for production of antibodies. Where do B cells mature? We have P type 1 and the B type 2. You should be able to explain that process. Then what are the important implications of the B cell defects? And this is what we have discussed. The Platons tyrosine enzyme, which is very important. When it is effective, then you have the Platons X-linked agammaglobulinemia, which is due to the fact that this enzyme is not properly expressed, and then somebody will have recurrent infections, and we have seen which type of infections. So you can read about the B cell ontogenesis on your own with more than one reference material, especially textbooks, and then you'll be ready for any question from the same. I want to stop there. I think we can discuss another time. Thank you so much.